Hello guys, welcome to my channel. So as you know, we are studying about the carbohydrates, right? So we are on the polysaccharide. So in the last couple of video we studied about the starch, glycogen, different polysaccharides, that's homopolysaccharide. So now moving further, as you know that when we studied about protein, like we studied about how the protein folds, right? So the same scenario like for the polysaccharide, what are the steric factors and what type of bonding that influences homopolysaccharide to fold. So the folding of polysaccharide in 3 dimension follows the same principle as the governing polypeptide structures, right? So it's the same kind of, so what is the same kind of thing if you remember in the polypeptide? So it's the subunits with a more or less rigid structure dictated by the covalent bond from 3 dimensional macromolecule structures that are stabilized by, so how they are stabilized? They are stabilized by weak interaction within or between the molecules, such as like hydrogen bonds interaction due to hydrophobic effect, van der Waals interaction, and for polymers with charged subunits, electrostatic interaction. So these are the things that helps in the folding of polysaccharide, right? Polysaccharide, I will write just here folding. And what influences? The same kind, right? The hydrogen bonds, hydrophobic effect, van der Waals interaction, right? For the ionic, it's the electrostatic interaction. So these are the mechanisms that same apply for the protein that applies here for the carbohydrates as well. So because polysaccharides have so many hydroxyl groups, right? As we know, polysaccharide has so many hydroxyl groups on their structure. So hydrogen bonding has an especially important influence on that structure because of the hydroxyl bonds. So glycogen, starch, and cellulose are composed of pyranoside subunits. That the pyranoside is a 6-membered ring subunit, as are the oligosaccharides as well, with the oligosaccharide like glycoproteins and glycolipids as well. So such molecules can be represented as a series of rigid pyranose rings connected by an oxygen atom bridging 2 carbon atoms. So the picture that you can see, that's what it explains. So like this is how pyranose ring is connected by an oxygen atom that bridges the 2 carbon atom, that is the glycosidic bond. So there is in principle a free rotation about the carbon and oxygen bonds linking residue, but as in polypeptide, rotation about each bond is limited by a steric hindrance by substituents, right? Right, and the most stable 3-dimensional structure for the alpha 1 to 4 linked carbon, right? From the alpha 1 carbon to the 4th carbon atom of the adjacent glucose molecule or monosaccharides molecule, linked chain of, that is the starch and glycogen, is a tightly coiled helix, right? So the picture you can see on the screen for the tightly coiled helix that is stabilized by interchain hydrogen bonds. And in amylose with no branches, right? The amylose has no branch. So this structure is regular enough to allow crystallization and thus determination of the structure by X-ray diffraction. Right, and for amylose, the core of the helix is of a precisely the right dimensions to accommodate iodine as a complex ions, like such as I3 and I5. So this complex can be found in the amylose helix, the iodine complex, right? This gives an intentionally blue complex to the structure. So this interaction is a common qualitative test for amylose. For cellulose, the most stable conformation is that in which each chair is turned 180 degrees relative to its neighbor, yielding a straight extended chain. So this you can see on the screen for the cellulose, the picture. So all the hydroxyl groups are available for hydrogen bonding with the neighboring chains, right? So with several chains lying side by side, a stabilizing network of interchain and intrachain hydrogen bonds produces straight, stable supramolecular fibers of great tensile strength. Right, so and this property of cellulose has made it useful to civilization for millennia. So like many manufactured products including paper, cardboard, rayon, insulating tiles, and a variety of other useful materials are derived from cellulose. And the water content of this material is low. Why? Because extensive interchain hydrogen bonding between cellulose molecules satisfies their capacity for hydrogen bond formation. So there is no room for water that can bind to the hydroxyl. Right, so that was a structure for the folding of homopolysaccharide. So there are bacterial and algal cell walls. So bacterial and algal cell walls that contain structures or structure heteropolysaccharide that contain structural heteropolysaccharide. So as we know, the rigid component of bacterial cell walls, that is peptidoglycan, right? So the rigid component of bacterial cell wall is what? Bacterial cell wall is peptidoglycan. That means carbohydrate and protein, right? So this is a heteropolymer, and as we studied in the Guyton or cellulose, right? So peptidoglycan heteropolymer, that is of alternating. What's the interaction between? So it's beta 1 to 4, right? The starch and glycogen was alpha 1 to 4. So this is beta 1 to 4 linked. And what kind of group? As it's peptidoglycan, so it is an acetyl glucosamine, and there is an acetyl muramic acid. Muramic acid, right? So these are the heteropolymers that lines the bacterial cell wall, right? So these are the linear polymers lie side by side in the cell wall. So this will be like something like this, like a linear polymer lie side by side, right? And they are cross-linked by a short peptides that are the proteins, right? And they are like cross-linked by a short peptide. This is how it might be linked. This is not the exact structure, but like just to understand. So they lie side by side, and they are linked by the cross-linked by the short peptides, and the exact structure actually depends on the bacterial species, what kind of bacterial species it is. So the structure depends on that kind of species, right? So the peptide crosslinks, so this crosslinks of the peptides weld the polysaccharide chain into a strong sheath, that is the peptidoglycan, right? That envelop the entire cell and prevents cellular swellings and lysis due to osmotic entry of the water. So this peptide, what it does, it prevents cellular swelling and lysis due to osmotic entry of the water, and this surrounds the entire cell, right? But the enzyme lysosome, right? Lysozyme, sorry. But the enzyme lysozyme, what it does, this lysozymes kill bacteria by hydrolyzing this beta 1 to 4 glycosidic bond. So this is a glycosidic bond in this kind of heteropolysaccharides, and this enzyme lysosome, this what it does, it hydrolyzes the beta 1 to 4 glycosidic bond between the acetyl glucosamine and N-acetyl muramic acid, right? And this enzyme is found in the human tears, right? This enzyme is found in human tears, lysozymes, where it is presumably a defense against bacterial infections of the eye. This is also produced by bacterial viruses to ensure their release from the host bacterial cell, and an essential step of viral infection cycle. So penicillin and related antibiotics kill bacteria by preventing synthesis of peptidoglycan crosslinks, leaving the cell wall too weak to resist osmotic lysis, and this is how the penicillin and related antibiotics kill the bacteria. So what the penicillin and other related antibiotics does, right? Penicillin and other related antibiotics, what it does, it prevents it prevents the bacteria by preventing the synthesis of this peptidoglycan crosslinks, this peptide crosslinks. It prevents this crosslinks, and if the crosslinks are not formed, so what will happen? The cell wall is too weak for the osmotic lysis, and the water can enter and it can burst, right? So that was the heteropolysaccharide, and acetyl glucosamine, and an acetyl muramic acid. So there is one more thing in the heteropolysaccharide, like certain marine algae, red algae, including some of the seaweeds, have cell walls that contains agar, right? So certain marine red algae and some seaweeds cell wall contain agar. What is this? A mixture of sulfated heteropolysaccharide. So this is a mixture of sulfated heteropolysaccharide, and this is made up of D-galactose and L-galactose. Made up of D-galactose and L-galactose, right? And these are derived and these are linked between C3 and C6, right? So this is 1 and 4, and these are linked between C3 and C6. So this agar is a complex mixture of polysaccharide, all with the same backbone structure, right? They are the complex mixture of polysaccharide, all with the same backbone structure, but substituted to varying degrees with sulfate and pyruvate, right? That's why it's sulfated, or it might be pyruvate. And agarose is the agar component with the fewest charge group. But what is the specialty? The remarkable gel forming property of agarose make it useful in the biochemistry laboratory. What's the specialty of this? Remarkable gel forming property of agarose, right? And this agarose is a agar component with the fewest charge group, so it has a very few charge group, and this has a gel forming property. How it is useful in the biochemistry laboratory? So when a suspension of agarose in water is heated and cooled, the agarose form a double helix, right? The two molecule in a parallel orientation twist together with a helix, repeat of 3 residues, and with helix what happens? The water molecules are trapped in the central cavity. So for example, these are the 2 molecule, and then after heating, like if they make like something like a helix, so in between here the water molecules are trapped in the central cavity, and this structure in turn associates with each other to form a gel, a 3-dimensional matrix that traps large amount of waters, right? And this structure, what it does in turn, associate with each other to form a gel like a structure, 3-dimensional gel like a matrix, that is a 3-dimensional matrix that traps large amount of water. So agarose gels, so this agarose gel, also called, are used as an inert supports for electrophoretic separation of nucleic acids. Agar is also used to form a surface for the growth of bacterial colonies, and the main another commercial use of agar is So what the capsules are made up of? They are made up of this agar, in which some vitamins and drugs are packed. The dried agar material dissolves readily in stomach and is metabolically inert. So the capsules are made up of the capsules that is made up of agar, the agarose gel, right? So that's it for this video. So I'll try to make a short one, but just you need to go and continue to watch the part 1, part 2, part 3. Right, so in this topic we just see the heteropolysaccharide and acetyl glucosamine and acetyl muramic acid, right, and the agar, right, and the folding of the homopolysaccharides. Alright guys, thank you guys for watching and we'll see you in the next video.